Welcome to part 7 of this video on the immune system. Okay, so we've now discussed the complement system, and therefore we've understood the purpose of the formation of an inflammatory exudate. What I now want to move on to is studying leukocytes in more detail. Remember, leukocyte is just a word for white blood cells. It's just the fancy way of saying white blood cells. Like erythrocyte is the fancy way of saying red blood cells. Some people will spell it with a C rather than a K. It doesn't matter. Okay, so what I want to start off with a more in-detailed discussion of is how are leukocytes actually recruited from the bloodstream into the extracellular fluid. So I want to start off with a more in-depth discussion of leukocyte extravasation. Then what I want to talk about is once we've actually got the leukocytes into the tissue fluids, the neutrophils and the macrophages, what are they actually going to do? The phagocytosis process, I want to discuss that in more detail and how that's going to lead to the destruction of pathogens that are present within our tissue. Okay, so let's begin then with the leukocyte extravasation process. And I do apologize, it's going to have to remain down here. I can't move this upwards, otherwise you know what happens. The camera makes everything on the page seem very faint, so I have to keep the paper down at the bottom here. Okay, right. So leukocyte extravasation. Well, remember back to the point where we were discussing type 1 and type 2 activation of endothelial cells. Remember that there must be some difference between what type 1 activation does to the endothelial cells and what type 2 activation does to the endothelial cells, because remember, type 1 activation only enables us to recruit neutrophils from the bloodstream into the tissue fluid, whereas type 2 activation allows us to recruit loads more neutrophils but also monocytes to differentiate into macrophages into the tissue fluid. So that's where I really want to go back to in order to discuss this. I want to go back to discussing type 1 activation. I want to discuss what change does type 1 activation cause in the endothelial cells that allows the endothelial cells to start recruiting neutrophils. That will allow us to discuss the actual process of the recruitment of neutrophils. And then what we'll move on to is discussing why type 2 activation then allows you to recruit monocytes and the different molecules needed to recruit neutrophils versus monocytes. Okay, so let's go back to type 1 activation then. So remember, type 1 activation is the very quick activation. Histamine will be produced, or released rather, from the mast cells very quickly following the detection of the pathogen and will act on the endothelial cells very quickly. So let's say that this is an endothelial cell that has undergone type 1 activation. Why is it now able to recruit neutrophils? Well, as part of the type 1 activation process, what it does is it fuses its viable pallade bodies with its apical membrane. So a really important concept now is the concept of the viable pallade bodies. Okay? And these are little vesicles stored in the endothelial cell. So I'll draw them like so, and they contain proteins that are going to be important for binding to things on the surface of the neutrophil as part of the neutrophil recruitment process. So this is going to represent a viable pallade body. Okay, so I'll draw a few more of them. So I'll have a few more here and here, so I'll colour them all in orange. Now let me just take a viable pallade body out and draw it bigger now. So I'll draw a bigger viable pallade body here. So here is the membrane of the viable pallade body, and in the membrane what you're going to have, like so, is a integral membrane protein known as P-selectin. So these proteins, which I'll colour in orange, these rectangles are representing integral membrane proteins that are stored in the viable pallade body and which are sequestered there and they're not in the apical membrane of the endothelial cells usually. But when the endothelial cell undergoes type 1 activation, it's going to fuse these viable pallade bodies with the apical membrane, and therefore these proteins are going to end up in the apical membrane. So I'll just write down the name of this protein. So these integral membrane proteins stored in the membrane of the viable play bodies are by the name of p-selectin, and I'll just write it out again because it's gone a little bit over there, so it's not particularly clear. So p-selectin, and it's another example of a lectin, a protein that is capable of binding to carbohydrates, and we will see the ligand for p-selectin in just a moment. So following type 1 activation then, so I'll put type 1 here, so following type 1 activation of the endothelial cell, what's going to happen is these viable play bodies are going to fuse with the apical membrane, and that's going to result in p-selectin, which was previously in the membrane of the viable play bodies, ending up in the apical membrane of the endothelial cells. And this is the change that now allows the endothelial cells to recruit neutrophils from the bloodstream into the tissue fluid. So let me now bring in a neutrophil. So I'll draw my neutrophil here. So the neutrophil has on its surface molecules that are going to be able to interact with p-selectin. So I'll just draw the nucleus of the neutrophil, the multi-lobed nucleus, like so. So this is our multi-lobe nucleus of the neutrophil, and on the surface of the neutrophil is going to be a molecule known as sialyl Lewis X, which is going to be able to interact with p-selectin molecules on the surface of the endothelial cells. So the next important player here is something by the name of sialyl Lewis X, which for short is often abbreviated down to S, and then you put LE for Lewis, so the S is for sialyl, LE for Lewis, and then you put the X up superscripted in this way. Now, this is the molecule that's going to be a ligand for p-selectin. Now remember, p-selectin is a lectin. It's going to bind to carbohydrate molecules. So sialyl Lewis X is actually a carbohydrate molecule. Specifically, it consists of 4 monosaccharides, and therefore it's a tetrasaccharide molecule. So tetra just means 4 saccharides. So it's a very small little carbohydrate molecule, and sialyl Lewis X is a tetrasaccharide which can be added on as a carbohydrate moiety onto a great many different molecules. So what I'm now going to draw here on the surface of the neutrophil, this is going to represent sialyl Lewis X. Now in reality, what you can really think of this, in fact actually I'll do it slightly better. I'll do it like this. So what I've done here is I've just drawn a line across there, and let's say that that little strip at the top there, that actually represents the sialyl Lewis X. And I do hope that's visible under these poor light conditions, but I've drawn a little green strip at the top there. The rest of that blob on the surface of the neutrophil represents some other integral membrane protein, or potentially glycolipid, so some glycoprotein or glycolipid which has the sialyl Lewis X attached to it. So here, this is the sialyl Lewis X. So all sorts of molecules on the surface of the neutrophil have this carbohydrate moiety attached onto them, which is the sialyl Lewis X, abbreviated like so. And this is the ligand for p-selectin. So, overall, what have we seen so far? So our neutrophil, here, and I'll just label him up. So our neutrophil is covered in sialyl Lewis X molecules, and it's these that are now going to interact with the p-selectin molecules on the surface of the endothelial cell, and this is going to lead to the endothelial cells being recruited from the bloodstream into the tissue fluid. So let me go over this in more detail. So actually the interaction between p-selectin and sialyl Lewis X is very, very weak, and they can form and break very easily. So what this actually plays the role in doing is in slowing the neutrophil down. And I'm just going to get a new piece of paper and then I'll show you what's going to happen. So a process known as rolling is going to occur. So the interaction between the p-selectin and the sialyl Lewis X is responsible for a process known as rolling, and this is exactly what it says. So if I draw the endothelium here, say this is the basement membrane and these here are the endothelial cells, and I should have done this slightly wrong. I would have liked a gap between those endothelial cells, but imagine there's a little bit more of a gap there, because of course the acute inflammatory response has occurred in this place, so the endothelial cells should have opened gaps in between themselves, between each other. So imagine there's a bigger gap there. Okay, but what's happened now is we know that once the endothelial cells have undergone type 1 activation, they'll have these p-selectin molecules on their surface, and I'll just draw these p-selectin molecules in here. So all of these can represent p-selectin molecules, and again I'll have them coloured in here in orange, like so. Okay, so now what's going to happen is the neutrophil is going to roll over the surface of the endothelial cells. So let's say here is our neutrophil. There's its nucleus. So what's going to happen is it's going to have interactions continuously forming and breaking between the p-selectin molecules on the surface of the endothelial cells and sialyl Lewis X molecules on the surface of the neutrophil. So there are the sialyl Lewis X molecules in green there. So that's sialyl Lewis X. And you're continuously going to be forming new ones and breaking new ones as the neutrophil rolls over the surface. So it's going to be going this way, rolling over the surface, continuously forming and breaking these interactions. And as these interactions continuously form and break, this will slow the neutrophil down. And this is actually the role of the p-selectin. It's not to form a strong adhesion with the neutrophil. It is just to slow the neutrophil down. So this is part one, if you like, of the leukocyte extravasation process. Whenever you're trying to move a leukocyte out of the bloodstream, the first thing you have to do is you have to get that leukocyte rolling along the surface of the endothelial cells and gradually slowing down. And this is what the p-selectin is going to do in the case of the neutrophils here. So it'll slow down as it does so. Right, so we put this p-selectin on the surface of the endothelial cells, and this allows the endothelial cells to have these neutrophils rolling along them to slow them down. Now, the next portion of the leukocyte extravasation process is what's known as tight adhesion. So once you've slowed the neutrophil down, what you can now do is form a very strong adhesion between it and the endothelial cells, and it is not p-selectin that is responsible for the tight adhesion. It is a different molecular interaction here, and the molecules responsible for the tight adhesion are actually already present on the apical surface of the endothelial cells. So the p-selectin is the only thing that we need to put on the surface of the endothelial cells so that we can slow the neutrophils down. We've already got the apparatus for performing the tight adhesion. It's just that it can't actually perform it because the neutrophils are moving too fast for it to occur. So if we now have another endothelial cell here, the endothelial cells have another important molecule on their surface, and this one is present there already. So type 1 activation doesn't need to occur in order to put this on the apical surface. It's already there. And this molecule is known as ICAM1, like so. And ICAM1 stands for intercellular adhesion molecule 1. So intercellular means between cells, and then it's an adhesion molecule, so it's going to form an adhesion between different cells. So intercellular adhesion molecule 1, ICAM1. And we'll have ICAM1 coloured in yellow here. Okay, so endothelial cells, they have this intercellular adhesion molecule 1 on their surface, and this is capable of forming a tight adhesion with a molecule on the surface of the neutrophils, provided that you already have the apparatus for slowing neutrophils down. If you haven't slowed neutrophils down, then this tight adhesion cannot form. So let me now draw my neutrophil here again, and once again I'll draw its multi-lobe nucleus here. So on the surface of the neutrophil you have a molecule, a protein, an intermembrane protein, an integral membrane protein by the name of LIFA1, LFA1, like so. So I'll colour LIFA1 here in green. So the neutrophils have on their surface a protein called LIFA1, and LIFA1 combines to the intercellular adhesion molecule 1 on the surface of the endothelial cells. Now, LIFA1 is always present on neutrophils, ICAM1 is always present on the endothelial cells. The reason these interactions don't usually form is that the neutrophils are moving too fast. In order for the interaction to form, you have to slow the neutrophils down. And in order to get the slowing process, you need this rolling process, and in order for that rolling process to occur, you need the p-selectin on the surface of the endothelial cells, and that doesn't arrive there until type 1 activation has occurred, which is why none of this occurs until you've had type 1 activation. So putting p-selectin there starts this entire process. So tight adhesion will now occur, and then once the tight adhesions are formed, and I should stress that you're not just going to form one of these interactions. You know, there will be loads of ICAM1 molecules on the apical surface of the endothelial cell, loads of Lympho1 proteins on the surface of the neutrophil. You're going to form lots of these interactions to form a strong, tight adhesion between the neutrophil and the endothelial cell. After that's occurred, the third part of the process is what's known as diapedesis. And diapedesis is the fancy name for the process where the leukocyte squeezes through the gaps between the endothelial cells, okay, under the guidance of molecules on the surface of the endothelial cells. So once this tight adhesion is formed, the endothelial cell is now going to help the leukocyte, in this case the neutrophil, to get between, get through the gap between the endothelial cells. So this is what it will look like. So here is the neutrophil, here's the neighbouring endothelial cell here, like so. And what's now going to happen is the endothelial cells are going to help the neutrophil to get across the endothelium, to get through the gap between the endothelial cells in this way. So here, once again, is the neutrophil's trilobed nucleus. Okay, so this process of a neutrophil deforming and squeezing through that gap between endothelial cells so that it can leave the bloodstream and go into the tissue fluid, this process is known as diapedesis. Okay, so after tight adhesion has occurred, the endothelial cells will coordinate diapedesis of the neutrophil through this gap between them. Okay, next what will happen is the neutrophil will now be in the tissue fluid outside the blood vessel, and then what needs to occur is it needs to migrate towards the actual site of infection. So what will now occur is migration, and the neutrophil, to do this, will just migrate towards the source of the pro-inflammatory cytokines. So it will go up the chemical gradients towards where there are higher levels of the pro-inflammatory cytokines, which is of course where they've been released from, and in that way it will find where the pathogen is present. Okay, so these are the major four parts of the extravasation of leukocytes: rolling, tight adhesion, diapedesis, and migration. We've discussed it in the context of neutrophils, but of course these same principles apply for the extravasation of any old leukocyte. Okay, so to summarize then, when the endothelial cells are type 1 activated by histamine, what will occur is the viable plate bodies will fuse with the apical cell membrane of the endothelial cell, putting the p-selectin integral membrane proteins on the apical surface of the endothelial cell. These p-selectin proteins, their ligand is obviously a carbohydrate because they're a lectin, hence the name selectin, and their ligand is a tetrasaccharide known as sialyl Lewis X, which neutrophils have all over their surface as a carbohydrate moiety attached to proteins and lipids. So what will happen is this will allow the neutrophil to continuously form and break interactions between sialyl Lewis molecules, sialyl Lewis X molecules rather, and p-selectin molecules on the surface of the endothelial cells, and this will allow the neutrophil to roll over the endothelial surface, and it'll gradually slow down as it's doing so. Then once it's slowed down, you can form tight adhesions between Lympho1, which is on the surface of the neutrophils, and the protein that's actually always present on the endothelial cells, known as ICAM1. Now, I'll stress again, the reason these don't usually form is the neutrophils are moving far too fast. You need the p-selectin on the apical surface to slow the neutrophils down. Once these tight adhesions are formed, diapedesis will then occur, which is the process by which the neutrophil squeezes its way through the gap between neighbouring endothelial cells and gets into the tissue fluid, and then once it's in the tissue fluid it just needs to migrate to the site where the infection is actually present, and it will just do that by migrating towards the source of the pro-inflammatory mediators. Okay, So what I now want to discuss is what's going to happen when the endothelial cells undergo type 2 activation in response to interleukin-1 and tumor necrosis factor alpha, because something changes to allow much more neutrophils to be recruited and also to allow monocytes to be recruited. So I want to now discuss that. Okay, so let's now discuss what's going to happen when an endothelial cell undergoes type 2 activation. So remember, type 2 activation, it takes much longer than type 1 activation, and hopefully you will remember the reason it takes much longer than type 1 activation. Firstly, it's because the mediators of type 2 activation are tumor necrosis factor alpha and interleukin-1, which, unlike histamine, are whole proteins, and therefore they have to be synthesized when the sentinel cell is activated, which takes time. But secondly, the reason is that once the tumor necrosis factor alpha and interleukin-1 have actually arrived at the basolateral side of the endothelial cell and are acting on receptors on the basolateral surface of the endothelial cell, and remember, hopefully you can remember the names of the receptors, tumor necrosis factor receptor type 1 and the interleukin-1 receptor type 1. Those receptors are going to activate complicated pathways inside the cell, which will lead to a change in gene expression, and that's the way in which these mediators change the behavior of the endothelial cells, by a change in gene expression. Whereas type 1 activation didn't, the changes in behavior there weren't brought about by changes in gene expression. So that's another reason that type 2 activation takes longer, because the changes in behavior of the endothelial cells that are type 2 activation require changes in gene expression to produce them. Okay, so what changes in gene expression then are you going to get which will allow this endothelial cell to recruit more neutrophils and also to start recruiting monocytes? Well, it's going to start synthesizing a protein known as E-selectin and putting that on its cell surface. It's also going to start producing more ICAM1. So let me explain this. So E-selectin is effectively equivalent to P-selectin. It's a different integral membrane protein, hence why it's got a different name, but effectively it does exactly the same thing as P-selectin. It's a lectin, and its ligand is the tetrasaccharide sialyl Lewis X, and we know that neutrophils are coated in sialyl Lewis X. So when you synthesize lots of E-selectin and put it on your apical surface, this is going to help with the rolling of neutrophils along the surface of the endothelial cells, because of course they will form weak interactions now between the neutrophil and the endothelial cell as well as the P-selectin was doing. Okay, so E-selectin is just bumping up our supply of these lectins on the surface of the apical membrane of the endothelial cell in order to increase the capability of the endothelial cell to have neutrophils rolling over its surface. So E-selectin, it's equivalent to P-selectin, not usually present, has to be synthesized. Okay, so that's one of the things that type 2 activation is going to do. It's going to put E-selectin on the apical surface of the endothelial cells, but utterly equivalent to P-selectin in function. ICAM1, we already know the function of that. That's for the tight adhesion of neutrophils. So hopefully you can completely understand why putting lots of E-selectin and lots of ICAM1 molecules on the apical surface of the endothelial cells are going to help with neutrophil extravasation. This is going to increase rolling of neutrophils, therefore we'll get more neutrophils slowing down. ICAM1 being bumped up is going to increase the amount of tight adhesion between endothelial cells and neutrophils, and therefore we'll get more diapedesis, more neutrophils recruited into the tissue fluid. So this is the reason then that type 2 activated endothelial cells are going to recruit far more neutrophils than type 1 activated endothelial cells, because we've bumped up the supply of the selectins and we have bumped up ICAM1 levels in the apical membrane of the endothelial cells. Now let's ask the question of how do we recruit monocytes, a new type of cell? Well, the principle of recruiting monocytes from the bloodstream, and I'll just draw a monocyte here, and thankfully monocytes are far easier to draw than neutrophils because they have a much simpler nucleus to draw. So the principle of recruiting a monocyte to the extracellular fluid is exactly the same as for neutrophils. You firstly have to slow the monocyte down, and you do this for a process of rolling. So how does rolling occur for monocytes? Is it different to rolling for neutrophils? And the answer is no. Monocytes too are absolutely covered in sialyl Lewis X. They have it attached to all sorts of proteins and all sorts of lipid molecules. So they too will roll because of the presence of these selectins on the surface of the apical membrane of the endothelial cells. So because of P-selectin and E-selectin on the apical surface of the endothelial cells, we are going to get monocytes rolling along the apical surface of the endothelial cells. Now hopefully at the moment you should be going, wait a moment, but surely that means that monocytes should have been recruited by type 1 activated endothelial cells, because when we had type 1 activation, those endothelial cells had P-selectin on their surface, so surely they would have got the monocytes to roll along their apical surface. And yes, you're right. The thing is, the type 1 activated endothelial cells don't have the necessary molecules on their apical surface in order to form tight adhesions between monocytes and the endothelial cells. So tight adhesion between monocytes and endothelial cells is different to tight adhesion between monocytes and neutrophils. It's different molecules, and I'm now going to discuss tight adhesion. So the principle is exactly the same. You have an integral membrane protein on the surface of the apical surface of the endothelial cells, and this integral membrane protein is going to be produced in response to type 2 activation, and this is by the name of VCAM1 rather than ICAM1, and this stands for vascular cell adhesion molecule 1. So vascular cell adhesion molecule 1. So I'll repeat, vascular cell adhesion molecule 1 is only going to be present on the apical surface of the endothelial cells once type 2 activation has occurred. When just type 1 activation has occurred, you won't have VCAM1 on the apical surface of the endothelial cells, and that's the reason that you can't recruit monocytes just with type 1 activation, because although you can get the monocytes rolling along the surface, you can't then form the typed adhesions, and that's useless. So only once type 2 activation has occurred, you have the necessary VCAM1 to form the tight adhesions, and then you'll actually get the full recruitment of monocyte process occurring. Okay, so VCAM1 is the integral membrane protein on the surface of the endothelial cells responsible for this tight adhesion. What's the protein on the surface of the monocytes then? This is by the name of VLA4 here. Okay, so monocytes have on their surface VLA4, which is important for forming these tight adhesions between the endothelial cells and the monocytes. So to go over this, to recruit a monocyte, you firstly need to have the rolling process. P-selectin and E-selectin can both do that, and that will bind to sialyl Lewis X tetrasaccharides on the surface of the monocyte. So you get rolling with type 1 activation, but you're not going to then get the tight adhesion, because for the tight adhesion you need VCAM1 on the apical surface of the endothelial cells to bind to VLA4 on the surface of the monocytes, and VCAM1 is not put on the surface of the endothelial cells in response to type 1 activation, and it's not just present in the way that ICAM1 is. So only once we've had type 2 activation of the endothelial cells will we get the expression of VCAM1 that's then going to allow us to complete the recruitment of the monocytes. What will then follow is diapedesis, just like this picture here, and then the monocytes will of course then differentiate into macrophages. That's the one process that you can add in here. The monocytes, once they've been extravasated, will turn into the macrophages, and remember I drew a picture of that over here. Here's a monocyte turning into a macrophage, becoming a huge blob of a cell, and then this macrophage will lumber its way towards the site of infection, i.e. migration. Okay, so that's the process of leukocyte extravasation. Remember, it requires the rolling process and the selectins binding to sialyl Lewis X, which is coating the neutrophils and the monocytes, is responsible for that. And then it requires tight adhesion, which for neutrophils is ICAM1 with LIFA1, and for monocytes is VCAM1 with VLA4. So this explains the reason that we get neutrophil recruitment following type 1 activation, i.e. the instant we've started the acute inflammatory response, we'll start getting loads of neutrophils appearing in the affected area, but then only later, after a few hours, will we start getting macrophages appearing, because that's when type 2 activation has come into play, and we've now got VCAM1 on the surface of our endothelial cells, which is needed for the recruitment of the monocytes. Okay, so we'll have a break here, and in the next video what we'll discuss is the phagocytosis process, i.e. we've got these neutrophils and these macrophages into our tissue now, what are they actually going to do to help?